Alternatively, we can take, instead of using the chemical Edman degradation method, we can also take our fragments and we can analyze them by using what's called tandem mass spectrometry. And so in this, what we want to do is we want to look at the masses of each of these fragments, and also we can fragment each of these fragments even more at the peptide bonds to get more information about the signature of these smaller peptide fragments, and that gives us a lot of information about what the sequence is. And so let's briefly discuss how mass spec works for proteins, so mass spectrometry, excuse me. So as you'll recall, probably from organic chemistry, so mass spectrometry can analyze molecules by their mass to charge ratio. Okay, and this is typically denoted by m over z. Okay, so when we have a protein, there are a lot of different ionizable residues. And so that can cause different charge states. And this is also helped by how proteins are introduced into the mass spectrometer. And so recall that proteins have hydration shells of water that surround them, that keeps them in solution. And so before a protein or even a peptide, which will also still have a hydration shell, can be introduced into the spectrometer, these waters need to be stripped. This hydration shell needs to be stripped. And so a technique called Electrospray Ionisation was developed. Ionization, or ESI, was developed in order to do exactly that. And so what this does is this basically sprays this protein solution like an aerosol into very tiny droplets, which are then very rapidly, because there's such a large surface area to volume ratio, then the water can be stripped very quickly. In fact, individual waters from the surface of the protein can be stripped very quickly from this, which allows the protein to enter now into the mass spectrometer, or the peptide fragment, I should say. So what that also does, though, is that tends to give you this distribution of charged residues. In particular, basic residues such as arginine or lysine tend to be in different states of protonation or deprotonation as this happens. And so what you get is a different charge distribution for the mass. So in the figure on the right, this is what's called a charge envelope. So this is a mass spectrum, or this is a spectrum from a mass spectrometer of apomyoglobin. And so this is in your book. And what you see is you don't actually see one peak for the mass of the entire protein, because the protein itself is about 16,960 daltons. But what you see is that there are several peaks. So there's one here at 1884.7, another one at 1696.3, 1542.3, and so on and so forth. And so what these actually represent are different charge states, because again, mass spectrometry separates by mass to charge ratio. So the charge is very important. So the mass, again, which is 16,960 daltons, is divided by the charge for each of these peaks. So for instance, the plus 10 peak is divided by 10, and so that indicates where the charge is 10, and so we see a mass of 1,696.3. Meanwhile, for instance, we can use another example, is 20, so the charge 20 plus So that's this mass divided by 20 instead. And so we see a peak at 848.7. And then there are varying states. So this is the same protein, just in different charge states. And so what we can do is, if we know the masses from these envelope, we can say, well, these are different charge states that vary by ±1. And then in practice, what we then can do is we can use that and back calculate for the mass if we don't know the mass of this of the protein or a peptide fragment if we're trying to identify the sequence of a peptide fragment.